Have you heard about Hatch and Slack cycle or C4 plants? And what exactly is the difference between C4 and C3 plants? You know, C4 plants are very efficient plants. What is the reason? Well, this is Dr. Pankaj Kumar, your mentor, and in today's sessions we will be discussing about C4 plants, and we will be discussing that why C4 plants is more efficient in comparison to C3 plants. So first of all, have a look at some of the basic features of the C4 plants. The perfect example of C4 plants are sugarcane and maize, right? And they are characterized by Cranch's anatomy. Now first of all, let's try to understand what exactly is a Cranch anatomy. You see, the meaning of the Cranch anatomy is that there are dimorphic chloroplasts, two types of chloroplast. Okay, one, they are going to have a chloroplast in a bundle seed, and second, there is going to have a chloroplast in a mesophyll. Okay, have a look at this diagram. You can see over there that this is the bundle seed. Okay, so in this bundle seed, what happens? They have a chloroplast, what we call bundle seed chloroplast, and these chloroplasts are known for having well developed dark reactions, or Calvin cycle we can say, right? But outside there is the mesophyll cells, okay? And in case of mesophyll cells, what we see that these are having a well developed the masonry that take care of the light reactions. So that's why we can say that there is a dimorphism in terms of the two different aspects of photosynthesis. One is the light reaction, second is the dark reaction. So in case of mesophyll, the masonry for light reaction is a well developed, but in case of bundle seed, the masonry for dark reaction is well developed. And it is this dual arrangement, right, what we call Cranch anatomy, actually give a great advantage to the plants in terms of the photosynthetic yield, the tolerance, and other benefit. We will discuss about that. But first of all, let's have a look at the cycle. As I told you that there exist two different chloroplasts. One we call mesophyll, second what we call bundle seed. So our cycle starts from the mesophyll cell, where the phosphoenol pyruvate, that is the 3-carbon compound, is going to act as a CO2 acceptor. Okay, so they accept a CO2 in the presence of enzyme what we call PEP carboxylase or pepcase, right? And oxaloacetic acid is formed. So this is the first stable product. Now the first stable product is a 4-carbon compound. That's why we call them a C4 cycle, right? Then this oxaloacetic acid gets converted into malate, and you know very well that the dehydrogenase enzyme is actually being utilized, and this malate is actually transported to the bundle seed cells. Okay, now within the bundle seed cells, what happens that the malate is actually decarboxylated. As a result of decarboxylation, the CO2 is being accepted by RUBP that is present in the bundle seed cells. So this RUBP carries on the Calvin cycle. Okay, and the another 3-carbon product, that is pyruvate, is formed, and that pyruvate is again transported back to the mesophyll cells. And in mesophyll cells, what happens that pyruvate by the dikinase activity. Okay, so this is what we call dikinase. By dikinase activities, with the help of 2 ATP molecule, again CO2 acceptor in the form of PEP is regenerated. Okay, so this is the general cycle what we observe in case of C4 plants. There are a few cases in which instead of malate, it is the aspartate that is transferred. So oxaloacetate gets converted into aspartate, and aspartate is again transported to the bundle seed cell, and aspartate again converted back to the malate, and rest of the reaction remains the same, right? So what is the key learning? The key learning is that there are only few things that can cross from mesophyll to bundle seed cells. Okay, and what are they? They are malate, they are aspartate, they are pyruvate. So these can cross, other molecules cannot cross. And what are the other advantages? Other advantages is that by this particular methods, the plant ensuring the greater concentration of carbon dioxide within the bundle seed cells, right? So that's why it leads to more and more carboxylase activity of the visco, leading to having more photosynthetic yield. So that is why the plant is more efficient in comparison to their C3 counterparts, right? Okay, so Again, you can see in the diagram. So this is how the cycle what we have seen. So this is the mesophyll. So you can see there. So for mesophills, again the CO2 is coming and the Calvin cycle is actually being prepared over there, right? And these are the steps. We know very well that CO2 is being accepted by PEP. So 4 carbon compound oxaloesterase is formed. It's converted into malic acid. Malic acid is transported into the bundle seed cell, and in some cases aspartic acid. And as far as bundle seed is concerned, we know very well that here what happens, malic acid is decarboxylated. So CO2 is actually going to be utilized in Calvin cycle, eventually forming the glucose, and pyruvate is transported back to the mesophyll where it generates PEP, that is CO2 acceptor, with the help of 2 ATP molecules, right? So this is all as far as C4 cycle is concerned. Let's talk about the energetics. Now you see here, for the fixation of 1 molecule of CO2, right, what is required? The requirement is 5 ATP and 2 NADPH2. Okay. Now out of this 5, what we see that 3 ATP is actually required in bundle seed during Calvin cycle. We know very well that in case of Calvin cycle, 3 ATP and 2 NADPH2 is required. Okay. Plus additional 2 ATP is required for dikinase activity. That is going to occur in case of mesophyll, right? So we can say altogether 30 ATP and 12 NADPH is required for the fixation of 1 molecule of glucose. And if you compare with the C3 plants, then additional, we can say that 12 additional ATP is required in comparison to the C3 plants, right? So this is the energetics what we know as far as the C4 plants is concerned. Okay. Right. So let's quickly see that what are the differences between C3 and C4 plants. The first thing, the CO2 acceptor. Okay. So in case of CO2 acceptor, you see that in Calvin cycle is a 5 carbon compound, but here it is a 3 carbon compound, right? First stable product is a 3-PZA, but here that is oxaloacetic acid. Chloroplast is monomorphic, here it is a dimorphic. In C3 plants, photorespiration occurs, but in case of C4 plant, photorespirations are negligible. Atmospheric carbon dioxide is not efficiently used because our Rubisco is undergoing the both carboxylase as well as oxygenase activity. But Cranch anatomy ensures that there is absolutely going to be carboxylase activities only. Okay. So it occurs in plants of all climates, but C4 plants are primarily observed in plants of tropical and subtropical plants only. So they require 18 ATP for synthesis of 1 molecule, but in contrary, it requires 30 ATP. Though energy is more required, but as far as the photosynthetic yield and output is concerned, it is far greater than the normal C3 plants, right? So what are the significance? The significance is that C4 plants have a competitive advantage over plant possessing the common C3 plants under conditions of what we can say, that is drought, high temperature and nitrogen and CO2 limitation. Okay. Secondly, the optimum temperature for C4 plants are 40 to 45 degrees Celsius, whereas for C3 plants is just 20 to 25 degrees Celsius. Okay. So here more carbon is fixed. Why? Due to absence of photorespiration. Okay. So altogether we can say that C4 plants, though they represent only 5% of the total global plants, but they accounts for 23% of the biomass accumulation. So you can understand that how efficient they are. As far as increasing the proportion of C4 plants on earth concerned, it is also going to help us in maintaining the balance of CO2, or we can accumulate more CO2, thereby we can free the atmosphere with the excessive amount of carbon dioxide in the atmosphere. So that is also one of the role of C4 plants. Okay. And C4 plants are an example of, perfect example of convergent evolutions. It is altogether 19 angiospermic families in which the C4 plants are present, but majorly it is represented in a monocots. And within the monocot, there are only 3 families. One, the Poaceae, second, the Chenopodiaceae, and third, that is Amaranthaceae. Okay. So these are the families which the C4 plants belong. So that is all what I have to say as far as C4 plant is concerned. So before we conclude, let's discuss one more important cycle, what we call CAM, or crassulacean acid metabolism. You know, CAM is just similar to the C4 plant. There's hardly any difference. The only difference is with reference to space and time. Let's have a look. First of all, the CAM plants are xerophytic plants. In them, what happens, stomats are going to open during the night, and in the day hours they remain closed just to check the transpirational loss of water. Okay, so the CAM metabolism helps plant to thrive at high temperature, intense sunlight and low soil moisture. Okay, have a look at the cycle. So what happens, CAM plants take CO2 through their stomats. They are accepted by the PEP, the phosphoenol pyruvate, the 3-carbon compound, forming the 4-carbon compound oxaloacetic acid as the first stable product. Now this is converted into 4-carbon malic acid, and they are stored in a vacuole called malate vacuole. Okay, so in the morning what happens, stomats are closed. So what happens, the accumulated malic acid undergoes decarboxylation, and as a result the CO2 that is formed due to decarboxylation, they are accepted by the RUBP in the Calvin cycle, forming the glucose, and again CO2 acceptor, the PEP is formed. So you see exactly it is the same. We can see over here. So in the night what happens, stomats open, the CO2 being accepted by PEP, forming oxaloacetic acid. Now they are converting into malate. The malate are being stored in a malate vacuole. During the day hours what happens, the malate get decarboxylated, CO2 is released and it is utilized in the Calvin cycle, starch is formed. Okay, then pyruvate, another, that is a 3-carbon compound, is formed. So pyruvate is again going to regenerate the phosphoenol pyruvate. Okay, exactly same. So here what we see, that your night period is actually equivalent to mesophyll and day period is equivalent to the bundle seed. So that's why it is absolutely similar to the C4 plants, right? What we see, that in C4 plants the carboxylation is going to occur in both day as well as night. Okay, have a look at this. So during the day hours carboxylation is happening as well as the night, both. But as far as the decarboxylation is concerned, okay, that is going to occur only and only during the day hours, when as a result of decarboxylation pyruvate is formed. So that is the things which is often asked in competitive examinations, right? So that's all as far as the entire C4 plants and CAM pathway is concerned. Thank you.